So for today, we'll be talking about microbial diversity. And for the first part of your lecture, we will be talking about acellular and prokaryotic microbes. So when we say acellular, these are this would include viruses, viroids, and prions. And for prokaryotic organisms, we will be talking about mostly bacteria. Okay? So there are actually several domains of bacteria. So this includes the true bacteria or the eubacteria and then the Archaea bacteria. Okay, so let's start our discussion. So microbes can be divided into the following. We've been discussing this since the introduction, whether they are considered as cellular. So when you say cellular, means that they are being provided with cell membrane. And the other one are those that are acellular. So we already agreed that those acellular bacteria do not have cell membrane, and we consider them as a non-biological entity. Okay. Now, cellular microbes can be divided into those that are prokaryotic. So when you say prokaryotic, again, let's review, means that they have a primitive nucleus. And then eukaryotic means that they have true nucleus because they have nuclear membrane. Prokaryotic do not have nucleus, but instead they have the so-called nucleoid. Okay. So prokaryotic includes bacteria and archaebacteria, while the eukaryotic organisms include algae, protozoan, and fungi. So viruses, viroids, and prions, even though they are acellular, they are still considered as infectious particles. So when you say infectious particles, they are still capable of causing infections. So they are still capable of causing infections. So that's the reason why we consider them as infectious particles. Okay. Now let's talk about viruses. Okay, so viruses are really very important because nowadays the reason why we're having a pandemic is because of a viral infection. So a complete virus particles, we call it virions. Okay, so it's like the basic unit. Okay, so virions. And when you say basic unit, it is already capable of infection. Okay, it's already capable of infection. So they are even much smaller than bacteria, between 10 to 30 nanometer in diameter. Or again, we utilize the term filterable agent for this. Okay? We utilize the term filterable agent for this. Okay. If a virus is capable of causing cancer, okay, so we call it oncogenic viruses, okay, or oncoviruses because they are capable of causing cancer. So an example of that is HPV, human papilloma virus, particularly the one that can cause genital warts. And so they are capable of causing cancer, or HTLV, human T lymphotrophic virus, can cause leukemia, for example. So they are capable of causing cancer. So a typical virion consists of either DNA or RNA. It's like this, no? Bacteria possess both DNA and RNA. So that's a typical characteristic of bacteria. But for viruses, viruses will possess only one type of nucleic acid. It's either DNA or RNA. So they never possess both nucleic acid. And why is it important? DNA or RNA, they are important because they are important for infectivity. Okay, so they are important for infectivity. Okay, so that's the reason why we consider them as important. Now, if we're going to draw a virus, so for example, this is the nucleic acid, which is either DNA or RNA. So the nucleic acid is being covered by a protein. Okay, so this protein is a capsid. We call it capsid. And the capsid is important because it gives the antigenic property of the virus. So when we say antigen test, we are detecting the viral proteins. And one viral protein is the capsid. Now, if we will be looking at this specific part, we can see a capsomer. Capsomer is the subunit of a capsid. So it is a subunit of a capsid. Okay, so that's the part, no? So I've shown you already the capsid and then the capsomer. So a while ago, I've drawn a virus and then I've drawn a capsid. And now some viruses are being covered by a lipid part. Okay, so this would be the envelope. Not all viruses have envelope. Okay, so it's actually made up of lipid and polysaccharides. The envelope is not coded by the viruses. It's actually buds off from the host cell. So hindi pa nagaling sa virus yung envelope, but an envelope of the virus actually buds off from the host cell. Okay, so viruses are considered to be as non-biological entities. And there are five reasons why we consider them as non-living things. Number one reason is that they possess either DNA or RNA. Other living cells such as humans, bacteria, possess both DNA and RNA. But for viruses, they can only possess one type of nucleic acid. Now, number two reason, they are unable to replicate on their own. Meaning to say, we consider them as an obligate intracellular parasite. So we consider them as an obligate intracellular parasite since they cannot replicate on their own. In fact, in order for you to cultivate viruses in the laboratory, you will be using a live tissue culture. So you will be needing a live tissue culture if you want to cultivate viruses in the laboratory. So unlike cells, they do not divide by binary fission because binary fission is for bacteria. Mitosis and meiosis is for the eukaryotic cells. So they do not divide by binary fission. They do not divide by mitosis or meiosis. but they divide by means of protein synthesis using the genetic machinery of the host. So that's the reason why viruses cannot really survive without a living host. So they lack the genes and enzymes necessary for energy production. So we also consider them as energy parasite. Okay? They depend on the ribosomes, enzymes, and metabolites of the host cells for protein and nucleic acid production. So that's the reason why, as what I told you before, they are important. Um, they actually rely on the genetic machinery of the host for protein synthesis. So this is how we classify viruses. So there are two major classification of viruses. We have the RNA virus, and then we have the DNA viruses. So the RNA viruses are divided into two categories. So those that possess envelope, we call them envelope RNA viruses. Those that do not possess envelope, we simply call them non-envelope RNA viruses. So non-envelope RNA viruses could either be single-stranded positive sense, ibig sabihin they have a single-stranded RNA, or a double-stranded RNA. So as regard with that of the envelope RNA viruses, they could be single-stranded positive sense, single-stranded negative sense, a retrovirus, linear virus, and segmented virus. So what is a retrovirus? A retrovirus has a special enzyme called reverse transcriptase, which allows them to produce DNA. Even if they are RNA, they can still produce DNA because of the presence of the reverse transcriptase. Okay? So these are examples of RNA viruses. So take a look at coronavirus, the one that causes COVID-19. Coronaviruses are classified as RNA virus. They are classified as envelope virus, and they are classified as single-stranded positive sense virus. Now, take a look at the DNA viruses. So DNA viruses could either be non-envelope and the other one is envelope. So under the non-envelope virus, we have the single-stranded linear virus, the double-stranded linear virus, and the double-stranded circular virus. And then under the envelope virus, we have a double-stranded linear virus, and the other one is the double-stranded circular virus. Okay, so this is how we classify viruses. Okay, so again, I think I've drawn this a while ago. So this is the nucleic acid, no? Nucleic acid can either be DNA or RNA. And why nucleic acid is important? They are important for the infectivity of the virus. So capsid is protein. They are also important for antigenic property. And each subunit of the capsid, we call it a capsomer. Okay, so since this virus do not have envelope, so they are known as the non-envelope viruses. So take note that non-envelope viruses are also known as the naked viruses. So they are also known as the naked viruses. Now here's an example of an envelope viruses, the structure of an envelope viruses. So we have here the nucleic acid. Okay, we also have the capsid and the capsomer, and now we have the envelope. Okay, so the envelope is actually lipid, and it actually not being coated by the virus, but instead it buds off from the host cell membrane. Okay, proteins in the envelope are also present. So sometimes we call it the spikes glycoprotein. If there are mutations in the spikes glycoproteins, that's the reason why there are some strains or variants of a certain virus, because it keeps on mutating. Hence, there would be a different appearance of the spikes glycoprotein. And this explains the reason why your coronavirus, after quite some time, will mutate and a new variant develops. So, as what I told you, the oops, baliktad lang, no? But anyway, the virus is actually, the envelope is actually not virus coded. As the virus is trying to leave the cell, it will take the cell membrane of the host. And once it has taken the cell membrane of the host, it will surround the entire capsid, allowing them to be an envelope virus. Okay, so it is not virus coded, but it came from the cell membrane of the host. Okay. So again, in summary, as what I've told you, viruses are classified. They are classified either DNA or RNA virus. So we can also classify them according to the shape and size of the capsid, the number of capsomer. So again, when say capsomer, we are referring to the protein subunit. Okay, and then of course, the presence or absence of an envelope. So we can classify them. We can classify them whether they are envelope viruses, or non-envelope virus, and then the disease it produces, no? So then the target cells, and then the immunologic or antigenic properties. So these are the different classifications of the viruses. So viruses, as what I've told you, or as what I've shown you in the graph a while ago, there are four major categories of the virus. Okay, most of the viral genomes are the first two types, the double-stranded DNA viruses and the single-stranded RNA viruses. So we also have single-stranded DNA viruses and double-stranded RNA viruses. So most viral genomes are circular molecules, again, but some of them are linear. Okay, so it is this is an electron micrograph, transmission electron micrograph view of herpes virus. So herpes virus is an example of an envelope virus. As you can see, before leaving the cell, it is still non-envelope. As this virus tries to leave the cell, it will take the cell membrane of the host, and then the cell membrane of the host will surround the entire capsid, and now you have a envelope herpes virus. So again, it is not coded by the virus, but it came from the host cell membrane. Okay, so these are the comparative size of the viruses. So as you can see here, it is an example of circular DNA virus. So bacteriophage, if you still remember the bacteriophage, the one that can infect bacteria. So it's very small. The poliovirus, the polyoma virus, adenovirus, the iridescent virus. So they are enumerated here according to the size. Okay, and then bacteriophage M13, the tobacco mosaic virus, bacteriophage T2. Chlamydia is not a virus, but it's a bacterium. And then we have the vaccinia virus. So imagine this is E. coli. So this is one bacterial cell, one bacteria, but viruses are even smaller than bacteria. The biggest virus, the Vaccinia virus, is even smaller than E. coli, just half of the E. coli, not even half of the E. coli. So that's how small they are. So viruses are so small. Hence, they are called filterable agent. They are able to pass through a bacteriologic filter. Okay, let's talk about bacteriophages, which I have already introduced to you last time. And this one is my favorite virus because majority of my researches are in bacteriophages. So bacteriophages are viruses that can infect bacteria. So hence we call them bacteriophages or symphyphages. And there are two types of bacteriophages. Okay, so the first one is called the virulent bacteriophages, and the second one is called the temperate bacteriophages. So when you say virulent bacteriophages, they undergo the life cycle known as the lytic life cycle. And what is the result of the lytic life cycle? The outcome of the lytic life cycle is the destruction of the bacterial cell. Okay? So it is actually visible as plaques. It is actually visible as plaques on the surface of the agar. So I think I've shown you the picture last time from our research. There are plaques in the surface of the agar. So it's actually a manifestation that the virus the bacteria were eaten by the viruses, literally eaten by the viruses. Okay? The other type is the temperate bacteriophages. Temperate bacteriophages will undergo the so-called lysogenic life cycle. Lysogenic life cycle. So when you say lysogenic life cycle, the DNA, the bacteria will not be destroyed, but the DNA of the virus is inserted into bacterial genome. Okay, so that's what will happen, no? And that insertion of DNA is called the provirus or the proviral DNA. Okay, so there are 5 steps in the lytic cycle. Okay, so we call it the attachment, penetration, biosynthesis, assembly, and release. Okay, so as you can see here, it's an electron micrograph of bacteriophages trying to infect the bacteria. Okay, so here's another example of a P2 bacteriophage inserting the DNA into the host cell. Okay, so this is an example of the lytic life cycle. So the first step is the attachment of the phage to the cell surface receptor, wherein the DNA is inserted into the genome. Okay, the second step is that the phage proteins, the bacteriophage will now start to synthesize new phage DNA and phage proteins. Okay, so synthesis is the third step. Fourth step is assembly. So new bacteriophages are formed. And number 5 is lysis, because this bacteriophage will now leave the cell and will now infect another cell. As they leave the cell, that causes lysis of the bacterial host cell. Hence, it's called lytic life cycle. Okay, so let's talk about animal viruses. A while ago, I was talking about bacterial viruses. But this time, let's talk about animal viruses. For animal viruses, we have several steps in the multiplication. So we have attachment, penetration, uncoating, biosynthesis, and assembly. Okay. So animal viruses escape from their host cell either by lysis of the cell. So if there is lysis of the cell, this can result to cellular destruction. And the other one would be by means of budding. Okay? So viruses that escape by budding becomes envelope viruses. I think I've shown you already the electron micrograph of such occurrence. Okay? So this is now an example of viral penetration. Okay? So this is an example if the virus is non-envelope virus. So the penetration will happen by means of endocytosis. Endocytosis is simple. It simply means that the virus will bind at the host receptor site, and as it binds, the invagination of the cell membrane will literally swallow the virus until such time that it is now inside the cytoplasm of the host cell, and the cell membrane a while ago becomes an endocytic vesicle. So you now have the virus inside the cytoplasm of the host cell. So this happens for non-envelope viruses. However, for envelope viruses, it's a different story. Because for envelope viruses, binding of a virus to a host cell membrane will cause fusion. Okay? There would be fusion of viral envelope with the host cell membrane. No invagination, but instead of invagination, there is fusion. Meaning to say, the virus will leave the envelope behind and only the capsid will enter the cell. So the envelope, okay, the envelope was left outside the cell. So hindi pala makapasok yung envelope, but only the nucleocapsid will enter the cell. Okay? So this is for the envelope viruses. Okay, I'll just be charging my device. Excuse me for a while. Okay. So I think you are familiar with this picture already, wherein the herpes virus is trying to leave the host cell, and as it leaves, the herpes virus is trying to penetrate the host cell, the HeLa cells, and as it penetrates the cell, so this is the envelope, no? This is the envelope. And then eventually, there would be what? Fusion. When you say fusion, the envelope of the virus would have conformational change. And that conformational change will allow it to be bind or fuse with the host cell membrane. And as you can see, only the capsid, okay, this is the capsid, only the capsid enters the cell. Okay, so only the capsid enters the cell. Okay, so that is for the envelope virus. Unlike in non-envelope, there would be endocytic vesicle because of the invagination of the cell membrane. But since we're talking about envelope viruses, there would be fusion of the viral envelope and with that of the host cell membrane. Okay, so let's talk about the terminology called latent virus infection. What do you mean by latent virus infection? It means, so the term latent, actually, in order for you not to forget it, just remember the word later. Okay, that's what we meant by latent. Because viral infections in which the virus is able to hide from the host immune system by entering the cells and remaining dormant. Okay, so perhaps one of the best example of latent infection is the one that can be caused by herpes virus. So herpes can cause cold sore, genital herpes, and chickenpox. Meaning to say, they never completely go away. They just become dormant. And you know, when a volcano is dormant, it simply means that it is inactive. It doesn't necessarily mean that it's dead already. Okay? So same like viruses, viruses could become dormant. Okay? It doesn't mean that they are dead. So that's the reason why once you have herpes, you'll always have herpes because they never completely go away. So for example, if you had chickenpox before, okay, so this is the neuron, the nerve fiber. So some of the viruses will simply hide inside the nerve fiber, wherein they become inactive for several years. So that's the reason why some people will have had chickenpox while they were young, and as they become adult, the virus becomes active once again, and it will reappear as shingles. So that's the reason why if I ask you, what is the latent infection of chickenpox? The answer is shingles. Even several years later, okay, shingles are actually the result of the same virus because they never completely go away. So that's what we meant by latent virus infection. Okay. Now, another important thing that you need to remember, antibiotics are not effective against viral infection. Meaning to say, if you have common colds, even if you drink antibiotics, antibiotics will not cure your viral infection. Antibiotics are antibacterial. So, in order to treat viral infection, you will be needing antiviral agents. Okay? So, antiviral agents are drugs that are used to treat viral infection. So, these agents interfere with virus-specific enzymes and virus production by disrupting critical phases in viral multiplication or inhibiting the synthesis of viral DNA, RNA, or proteins. So meaning to say, the DNA, RNA, and proteins, okay, of the viruses will be affected, okay, if you will be drinking or if you will be treating yourself with antiviral agents. Okay. So another terminology that you need to remember, okay, oncogenic viruses or oncoviruses. So when you say oncogenic viruses or oncoviruses, means that these viruses are capable of causing cancer. Okay, so I have included here the Epstein-Barr virus. Okay, so the Epstein-Barr virus, it is related to Burkitt's lymphoma, the human papilloma virus, particularly the type 16 and type 18 can cause cancer of the cervix. Penile cancer, because this yung human papilloma virus that can cause cancer are the genital warts. and then the human T lymphotrophic virus, which a while ago I had mentioned that can cause leukemia. Okay. So another example of important virus is the HIV, the virus that causes AIDS or acquired immunodeficiency syndrome. It is an enveloped single-stranded RNA virus, and I think I've already mentioned the term reverse transcriptase is the enzyme that allows the RNA to code for the complementary DNA. Now, why do you think it causes acquired immunodeficiency syndrome? It's because it targets on CD4 cells. CD4 are the T helper cells. And then these T helper cells are important in humoral immunity and even in cell-mediated immunity. Okay, so normally we have between 800 to 1,000 times 10 to the 9 per liter of CD4. However, once your CD4 level falls below 200 times 10 to the 9 per liter, you will have now AIDS. And these AIDS are characterized by the presence of various opportunistic infections. So when you say opportunistic infections, these are infections caused by organisms that take advantage of hosts with weak immune system. Okay, so if your CD4 level falls below 200 times 10 to the 9 per liter. Okay, and one of the most important enzyme is the reverse transcriptase that allows them to code for the DNA. So we also have the viroids and the prions. Okay, so the viroids and the prions are smaller and less complex infectious particle than viruses. Okay, so yung prions, I think you are more appropriately termed as infectious proteinaceous molecule. Causes mad cow disease, for example. Viroids are a short naked fragment of single-stranded RNA which can interfere with the metabolism of plant cell. So therefore, they are considered to be as plant pathogens. So the viroids are transmitted between plants in the same manner as viruses. So example of plant diseases caused by viroids are potato spindle tuber and the citrus exocortis. So these are examples of plant diseases caused by viroids. Prions are even smaller infectious proteinaceous molecules that causes fatal neurologic disease in animals and in humans. So in animals, it can cause scrapie in sheep, bovine spongiform encephalopathy in cows. It is more commonly known as the mad cow disease. And in humans, if we eat the contaminated meat of infected cow, we can still get prions even if we cook it thoroughly. And in humans, it's called Creutzfeldt-Jakob disease. Okay? Of all pathogens, remember this one. This one is very important. Prions are the most resistant to disinfectants. So the mechanism by which prions cause disease remains a mystery. Okay, so that's prions. Okay, so I have already discussed the first domain, which is about viruses. This time, I will be talking about the bacteria, the domain bacteria. Okay, bacteria are divided into three major phenotypic categories. Okay, gram-negative, they have a cell wall. Gram-positive, they also have a cell wall. And those that lack cell wall, we call it mycoplasma. Okay, so mycoplasma. The characteristic of bacteria is used in classification and identification. So this includes cell morphology. So we can classify whether they are cocci or bacilli. Staining reactions, we classify them whether they are gram-positive or gram-negative. Motility, we classify them whether they are motile or non-motile, depending if the flagella are present. Colony morphology, atmospheric requirements, nutritional requirements, biochemical and metabolic activities, enzymes that the organism produces, pathogenicity, and genetic composition. Okay, so let's discuss them one by one. Okay, so there are three basic shapes, okay, or basic categories of bacteria based on the shape. Cocci are actually plural, coccus is the singular form. So this is a coccus, and these are the cocci. Okay, so bacillus is the singular form, and there are several of them. We call it the bacilli. And then there are even curved or spiral-shaped bacteria. So the curve is usually called vibrio. The spiral-shaped bacteria, spirillum. Okay, so spiral-shaped bacteria. Cocci may be seen singly or in pairs. We call it diplococci. Or in chain. We call it streptococci if they are in chain. Or in clusters such as this, we call it staphylococci. Or tetrads. Or packet of 8 or octads. Or sometimes we call it sarcinae. We call it sarcinae or the cubical pocket of 8. Okay, so the average coccus is about 1 microns in diameter. Some cocci have coccus in their name. So for example, we have Staphylococcus or Streptococcus. Okay. So again, we have here the cocci, curve-shaped and spiral-shaped bacteria, and then we have bacilli. So diplococci are actually seen in chain. Diplococci are actually seen in pairs. So we have Neisseria gonorrhoeae, and Neisseria gonorrhoeae is the causative agent of gonorrhea. We have Streptococci, which is in chain. An example of is Streptococcus pyogenes that can cause strep throat. Staphylococci are cocci in clusters. So our example here is Staphylococcus aureus, and Staphylococcus aureus, it can cause boils, pigsac. And then tetrads. So our example here is Micrococcus luteus. It's rarely pathogenic. And a packet of 8 is Sarcina ventriculi, which is also rarely pathogenic. So here is an electron micrograph view of Staphylococcus. And here is the gram stain. So for the gram stain, we consider them as a gram-positive bacilli. Okay, so let's talk about the bacilli. Bacilli are often referred to as rods. They may be long, short, or long, thick or thin, pointed or with curved or blunt ends. They could appear singly, so that's a bacillus, or by pairs, diplobacilli, or by chains, so we call it streptobacilli, in long filaments or branch. An average size bacillus is 1 micron in width, 3 microns in length. If the bacilli are extremely short, we call it a coccobacilli, which is a transition between a coccus and a bacilli. Examples of medically important bacilli are Escherichia, Klebsiella, Proteus, Pseudomonas, Haemophilus, and Bacillus species. So these are some of the medically important bacilli. Okay, so we also have the curved and the spiral-shaped bacteria. So Vibrio, Campylobacter, and Helicobacter is the example of curved bacteria. So noteworthy to mention is Vibrio cholerae, which can cause cholera. Then our examples of spiral-shaped bacteria are Treponema. So Treponema pallidum, if you're familiar with this one, causes syphilis. Borrelia burgdorferi causes Lyme disease. Okay, so these are examples of medically important spiral-shaped bacteria. Okay, so here's an example of Treponema pallidum. Borrelia hermsii, another example of spiral-shaped bacteria that can cause relapsing fever. So that's Borrelia hermsii. Okay, aside from these, we also have star-shaped bacteria. We call it the stella. And then the rectangular shaped bacteria is the Haloarcula. So Haloarcula. Haloarcula is an example of rectangular shaped bacteria. Okay, so the three major categories of staining procedures are the simple staining procedure, the structural staining procedure, and the differential staining procedure. So for the simple staining procedure, it uses one dye. Means that whatever the color of the stain will be the color of the bacteria. So if you're using methylene blue, then the color of bacteria would be methylene blue. Then the structural staining procedure is actually used for special parts such as the capsule, spore, and the flagella. Now, the differential staining procedure will divide, bacteria into two major categories. For example, if you are using gram stain, the two categories are gram positive, then gram negative. If you are using acid fast stain, the two major categories are acid fast positive and acid fast negative. So these are the two major categories. Okay, so while it's in staining, bacterial smears must be fixed prior to staining. So for example, this is the slide. You place the smear here, and then you fix it over the alcohol lamp. So you have an alcohol lamp here, and then you place the slide here so that it will be fixed. Okay, so this is what you call heat fixation. So heat fixation is not a standardized technique, and the major disadvantage of that is that it will distort the bacterial morphology. So the preferred technique is the methanol fixation. So which means that we simply add the alcohol methanol. So that is called fixation. Why fixation is important? Fixation is important because the process will kill the organisms, so it will be safer for us to study them, and then it will preserve their morphology and anchor the smear to the slide, so that once we treat them with different kinds of stains, they will not be washed away because they had already been fixed, okay, onto our slide. So that's the importance of fixation. Okay, so here is the procedure for a simple stain. So all you have to do is to place the smear into the slide and allow it to air dry. And then we add methanol, okay, to fix. And then we flood the slide with the stain. So for example, this is a methylene blue. So whatever the color of the stain is, will be the color of the bacteria. So after flooding it with methylene blue, we rinse it with water and blot it with air dry, and then we examine it under the microscope. Okay, so this is a simple bacterial staining technique. Okay, and then another important, I think the most important staining technique in microbiology is the gram staining. Okay, so in your lobster, you've been doing dissimulation, and of course, it's very important that we know the principle. So gram stain will allow the bacteria to be classified as gram positive or gram negative. Okay, so gram positive bacteria end up being blue or purple. Gram negative bacteria end up being pink or red. So the final gram reaction, whether it's positive or negative, depends on the cell wall structure. So last week, I have already mentioned to you that gram-positive bacteria have a thick layer of peptidoglycan, which will make it difficult for the crystal violet iodine to be decolorized because of a very thick peptidoglycan. Gram-negative bacteria, on the other hand, have thinner layer of peptidoglycan, which will allow crystal violet to be washed away, to be decolorized, and the cells will subsequently take up the safranin, which is pink or red in color. So this is the basic difference, no, between the gram-positive bacteria and gram-negative bacteria. The final color of gram-positive bacteria is blue or purple. The final color of gram-negative bacteria is either pink from pink to red. The peptidoglycan for gram-positive bacteria is much thicker, which will not allow crystal violet to be washed away easily. Unlike gram-negative bacteria, they have a thin layer, a much thinner layer of peptidoglycan, which will not hold crystal violet as much as the gram-positive, hence crystal violet will be washed away. So teichoic acid and lipoteichoic acid is found in gram-positive bacteria, but not in gram-negative bacteria. LPS, or the lipopolysaccharide, is not found in gram-positive bacteria, but it is found in gram-negative bacteria. Okay, so this is now the procedure. So we place the smear into the slide and we add methanol. So what's the purpose of methanol? Fixative. And then we add crystal violet. Okay, so take note ha, crystal violet is a primary stain. Okay, crystal violet is the primary stain. Okay, so we rinse the slide, and after rinsing the slide, we flood the smear with iodine solution. What is the purpose of iodine? Iodine is a mordant. Okay, what is a mordant? The mordant will intensify the crystal violet iodine complex. Okay, initially both gram-positive and gram-negative will be purple. Upon the addition of crystal violet and iodine, both gram-positive and gram-negative will initially be purple. And then we rinse off excess iodine, and then we decolorize it with an ethanol, particularly the 95% ethanol or ethyl alcohol, for about 5 seconds. So what's the purpose of 95% ethanol? It serves as decolorizer. So ethanol will serve as decolorizer. So what will happen upon adding ethanol? Upon adding ethanol, okay, gram-positive bacteria remains purple. Gram-negative bacteria becomes colorless. So upon adding 95% decolorizer, this is what will happen. Okay? Gram-positive bacteria remains purple. Gram-negative bacteria becomes colorless. Why? Why do you think gram-negative bacteria becomes colorless? Because they were decolorized. They were decolorized, hence becomes colorless. And then after that, we add safranin. So you wash immediately the slide in water, and after organisms, after washing, so it remains purple. Gram-negative bacteria becomes colorless. Why they are colorless? Because they were decolorized. Okay, and then we add safranin. Safranin is the counter stain. So what is the primary stain? The primary stain is crystal violet. And what is the counter stain? The counter stain is safranin, which is between pink to red in color. So after washing with water and air blot it, we can now look under the microscope. So if it's purple, or blue to purple, it's gram positive. But if it's red to pink, then it is gram negative. Okay, so that's how we perform the gram stain in the laboratory. And this is how they look like under the microscope. So you'll be able to see an example of gram-positive bacteria. This one is dark blue, no? So basta between blue to purple. So they are considered to be as gram-positive. So another example of gram-positive, Clostridium perfringens. Okay, Clostridium tetani is another example of gram-positive bacteria from blue to purple. Okay, so it's another example of gram-positive bacteria. Now, these are examples of gram-negative bacteria. So they appear to be red to pink in color. So if they appear red to pink, then they are gram-negative. Okay, so there are various bacteria that can be observed in gram stain. Okay, so what are the rules in gram staining? All cocci are gram positive, except for Neisseria, Veillonella, and Branhamella. All bacilli are gram negative, except for Bacillus. Bacillus is a gram-positive bacteria. Corynebacterium. Corynebacterium is a gram-positive bacteria. Mycobacterium. So Mycobacterium is a gram-positive bacteria. Nocardia. It's a gram-positive bacteria. Erysipelotrix. It's a gram-positive bacteria. Lactobacillus. It's a gram-positive bacteria. Listeria. It's a gram-positive bacteria. Pursia. It's another example of gram-positive bacteria. Rothia. This is H. It's another, so these are gram-positive bacteria. So the rest of the bacilli are considered to be as gram-negative. So all bacilli are gram-negative except for Bacillus, Corynebacterium, Mycobacterium, Nocardia. I forgot another one, Clostridium. Nocardia, Erysipelotrix, Lactobacillus, Listeria, Pursia, Rothia. Okay, so these are the rules in gram staining. So gram staining is one of the most important kind of staining here in microbiology because we'll be able to define whether the bacteria are gram-positive or gram-negative. Okay. Some bacteria are neither consistently purple nor pink after the gram stain. They are considered to be as gram variable. So, though Mycobacterium are gram-positive, but it is not the best staining method for Mycobacterium because of the fact that Mycobacterium has mycolic acid. So Mycobacterium has mycolic acid, which is lipid in nature and waxy, hence making it difficult for crystal violet to penetrate the cell wall of Mycobacterium. Therefore, instead of using gram stain for Mycobacterium, we should be using the acid-fast stain. Okay? So, in the acid-fast stain, carbolfuchsin is the red dye that is driven to bacterial cell wall using heat. Okay, so carbolfuchsin here serves as the primary stain. So carbolfuchsin serves as the primary stain. And the decolorizer in acid-fast stain is acid alcohol. Hence the term acid-fast. Okay? So what do you mean by acid-fast? Okay, so the term acid-fast refers to ability to resist decolorization by acid alcohol due to the presence of mycolic acid. So acid-fast bacilli are actually red because of the carbolfuchsin. Non-acid-fast bacilli are actually blue because of the methylene blue as counter stain. So as you can see here, these are example of acid-fast bacilli. So what are the rules? So the rules are all bacteria are non-acid-fast except for Mycobacterium. So all bacteria are non-acid-fast. All bacteria are non-acid-fast except for Mycobacterium. And then Nocardia is slightly acid-fast. So Nocardia is slightly acid-fast. Okay, so these are the rules in acid-fast state. Okay, so moving on, we also classify bacteria whether they are motile or non-motile. So if a bacterium is able to swim, okay, swim, it is said to be motile. So the bacterial motility is associated with flagella, and less often with eggshell filament, because eggshell filament is the endoflagellum. Most spiral-shaped bacteria and about 50% of bacilli are motile. Cocci are generally non-motile because they do not possess flagella. So motility can be demonstrated by stubbing the bacteria in a tube of semi-solid medium. So the most common example of a semi-solid medium is SIM, which stands for sulfide indole motility. So here you will be using an inoculating needle, and then you stab the needle with bacteria into the medium, into the semi-solid medium. And a positive result is characterized by growth away from the site of inoculation. So if there is a growth away from the site of inoculation, then that is a positive result. So something like this. There is a growth away from the site of inoculation, then that is positive for motility. Okay, another one is using hanging drop technique. This one is much easier because all you have to do is to add a drop of bacterial suspension into a cover slip and then add petroleum jelly on the site, and then you place a slide on top of it and then reverse it, then look under the microscope so that you'll be able to see motile or moving organisms. Okay, so that is for the hanging drop technique. Okay, so that we'll be able to know if the organisms are motile or not. So here is an example of a negative result because there is no growth away from the site of inoculation. And here is an example of a positive result because of the growth away from the site of inoculation. Okay, so that is semi-solid medium. And here is the hanging drop technique. So the hanging drop technique is all you have to do is to add a drop of bacterial suspension, living bacteria, and then you add cover slip with petroleum jelly into a concave slide. So you have to have a concave slide in order for you to see the hanging drop technique. Okay. So if the organisms are able to grow in a culture or in the petri dish, we call it the colony of bacteria. Excuse me for a while. So here you'll be able to see bacteria that can be grown in the petri dishes. Okay, so the colony morphology or the appearance of the colony varies from one species to another. So it depends, no? It varies. So the variation includes the size, the color, the overall shape, and even the elevation and appearance of edge or margin of the colony. So later on, I will be showing you a petri dish of our bacterial culture here in the laboratory. So colony morphology can also include the results of enzymatic activity on various type of media. So sometimes we call this biochemical testing. So we can perform biochemical tests in the culture media. As is true for cell morphology and staining characteristic, colony morphology is an important clue to identify the species of the bacteria. So it is not conclusive, but you'll find clue. So I have here an example of a petri dish. So this is a petri dish. This is where we culture the bacteria. So I hope you can see. This is how the bacteria look like. No? So you have culture of bacteria. So this is what there are millions of colonies, and each colony will contain million of bacteria. So we have here another example of E. coli growing in McConkey agar. And so I hope you can see them. And so maybe I can place it in display again in the lab. So if you have time, you can pass through the lab to observe how they look like. No? So that's how we culture bacteria. So we culture bacteria in culture media. Okay? So the size of the colonies is determined by the organism's generation time, and is another important characteristic of a bacterial species. So here's the generation time that is assumed to be 30 minutes. What does it mean? Means that the bacterial population doubles its population every 30 minutes. So for example, this is a single bacterial cell. So the number of cells is just one. After 4 hours, because of the generation time and it's a logarithmic increase, the bacterial cell becomes 256 and the colonies are much bigger, but it's still not yet visible. After 8 hours, the bacterial cell is about 65,000 and the colonies are not yet visible. But after 12 hours, the colonies becomes visible. And each colony, this is just one colony, contains about 17 million bacterial cells. Okay? So that is the size of the colonies, no? So which means that the bigger the colony, then the older the bacterial cells are. Okay? So bacteria can be classified also on the basis of their atmospheric requirements in relationship to the oxygen and carbon dioxide requirements. So with respect to oxygen requirements, we can classify them as obligate aerobic, microaerophilic aerobic, facultative anaerobic, aerotolerant anaerobic, and obligate anaerobic. Okay, so when you say obligate anaerobic, obligate means that they are strictly anaerobic, meaning to say oxygen is toxic for them. So oxygen is toxic for them. They cannot survive in the presence of oxygen, such as Clostridium. Clostridium is an example of anaerobic bacteria. So they will not be able to survive in the presence of oxygen, which is the exact opposite of obligate aerobic. Obligate aerobic means that they are strictly aerobic. They need oxygen. They need oxygen. So the example of this one is Mycobacterium Tuberculosis, the bacteria that can cause TB, and they reside in our lungs. And our lungs are very rich in oxygen. Okay, so why do you think they are able to survive oxygen? Because of the enzyme system. Okay, there is an enzyme system that allows bacteria to make the oxygen toxic into a non-toxic form. So these enzymes are super oxide dismutase, catalase, and peroxidase. So these are the enzyme system that will make the toxic form of oxygen into a non-toxic form. And mostly, obligate aerobic bacteria have this enzyme system in which this enzyme system is not present for anaerobic bacteria. Okay. How about aerotolerant anaerobes? So when you say aerotolerant anaerobes, they are fundamentally anaerobic but can tolerate oxygen. So they can tolerate oxygen. Perhaps they have only one enzyme system. Okay? Facultative anaerobic, these are fundamentally aerobic but can survive in the absence of oxygen. That's facultative anaerobic. And then we have the microaerophilic. The microaerophilic needs only a small amount of oxygen. Okay, so they would only need a small amount of oxygen. Okay, so that's how we classify them. Now, in relation, with respect to carbon dioxide requirement, we classify organisms as capnophilic. Capnophilic grows best in the presence of increased concentrations of carbon dioxide, usually between 5 to 10%. So usually we culture the bacteria with CO2 packets or candle jar. So this is a thioglycolate broth that can support all kinds of organisms. So usually aerobic organisms can grow here because this one is the most exposed surface that would have about between 20 to 21% of oxygen. Microaerophilic will grow here because this one would only have small amount of oxygen, and obligate anaerobe will grow here because the bottom doesn't have oxygen. Okay. As regards to the nutritional requirements, all bacteria require some form of elements such as carbon, hydrogen, oxygen, sulfur, phosphorus, and nitrogen for growth. So some bacteria would require special elements such as calcium, iron, or zinc. Organisms with special demanding nutritional requirements are said to be fastidious, fussy, or demanding bacteria. So usually we grow them in blood agar plate. The nutritional needs of a particular organisms are usually characteristic for that species and are sometimes important clues to its identity. Okay? So as bacteria grow, they produce many waste products and secretions, of some of which are enzymes. So pathogenic strains of bacteria such as Staphylococcus or Streptococcus can be tentatively identified by the enzymes they secrete. So that can be determined also in the laboratory. In particular environments, some bacteria produces gases such as CO2 or H2S or hydrogen sulfide. So to identify bacteria in the laboratory, they are being inoculated in various substrates or biochemical culture media to determine whether they possess the enzymes necessary to break down those substrates. So many pathogens are able to cause diseases because they are because of the virulence factor, which I have already discussed from last week, no? So this includes the capsule, pili, or endotoxin. So, and some of them can even secrete exotoxin or exoenzymes that can damage cells and tissues. Frequently, the pathogenicity is tested by injecting the organisms into the mice or cell culture. So we call it the animal virulence test, or sometimes we culture living cells with bacteria and to determine the effects of bacteria in those living cells in vitro or in the laboratory. So example of pathogenic bacteria, Mycerium meningitidis, which can cause, this one can cause meningococcinia. Salmonella typhi can cause typhoid fever. Shigella can cause bacillary dysentery. Vibrio cholerae, obviously, this one can cause cholera. Yersinia pestis, this one causes the Black Death in the medieval ages. Treponema pallidum can cause syphilis. So these are all examples of pathogenic bacteria. Laboratory identification of bacteria is moving toward analyzing DNA or RNA called molecular diagnostic procedures. So that's the reason why PCR has become very important technique in identification of the bacteria. So the composition of the genetic material or the DNA of an organism is actually unique to each species. So that's why I told you during the introduction that DNA is like the blueprint of life, not just of the bacteria, but even of our life as humans. So DNA probes make it possible to identify and isolate without relying on phenotypic characteristic. Okay? Though the use of 16S RNA sequencing through the use of the 16S RNA sequencing, the degree of relatedness between two different bacteria can be determined. Now, take note, when we say 16S RNA, we are referring to prokaryotic cells. But for eukaryotic cells, we will be referring to 18S RNA sequencing. Okay? There are also some bacteria that are called unique bacteria. Okay? Rickettsias, Chlamydias, and Mycoplasmas are bacteria because are unique bacteria because they do not possess all the attributes of a bacterial cell. So Rickettsia and Chlamydia are gram-negative type of cell wall, but they are considered as obligate intracellular parasite because they cannot be grown in a culture media. In fact, Rickettsia have leaky membranes. Chlamydia naman cannot be grown on artificial culture media because they are energy parasites, meaning to say they need or they prefer to use the ATP of the host cells. So they are like virus, but they are not virus. They are prokaryotic cells. Mycoplasma, on the other hand, are the smallest of the cellular microbes. So they lack cell wall, therefore they assume a variety of shape. So they are known as pleomorphic. In fact, we classify mycoplasma as PPLO, which stands for pluro pneumonia-like organisms. In humans, pathogenic mycoplasma causes primary atypical pneumonia. So this one is sometimes known as the walking pneumonia and genitourinary infections. Because they have no cell wall, they are resistant to drugs like penicillin. Because if you drink penicillin, penicillin will attack the cell wall. However, in the absence of the cell wall, there's nothing penicillin will attack to. So therefore, penicillin will be useless for them. So mycoplasma can produce a tiny fried egg appearance on artificial media, such as the one that you can see here. So let's talk about photosynthetic bacteria. So photosynthetic bacteria includes purple bacteria, the green bacteria, and the cyanobacteria or the blue-green algae. So they all use light as an energy source, but not in the same way. So for example, purple and green bacteria do not produce oxygen, but cyanobacteria do. Now, photosynthesis that produces oxygen is called oxygenic photosynthesis. However, if the photosynthesis does not produce oxygen at all, we call it anoxygenic photosynthesis. Okay, so that's for photosynthetic bacteria. Now let's talk about the Archaea. So the term Archaea means ancient, and they were just discovered in 1977, and they are still considered as prokaryotic organisms. Now, genetically, Archaea are more closely related to eukaryotes than to the bacterial counterpart. And Archaea vary widely in shape, and they live in extreme environments, extremely acidic, extremely hot, or extremely salty environments. So Archaea possess cell walls, but the cell walls do not have peptidoglycan. Unlike the true bacteria will have peptidoglycan in the cell wall. Okay? So if you still remember, we classified the Archaea bacteria as the methanogens because they can produce methane gas, the extremely halophilic, and then the extremely thermophilic Archaea bacteria. Okay, so that ends our first lecture for today.